Hi, everyone. I know more people are going to be piling in the next few minutes, but we're going to get ahead and get started. So just to introduce myself and everyone, I'm Annetta. I'm one of the interns here at Bridge. And then we have with us Dr. Julia Plank and Dr. Tamar Green, who will be presenting today. Um, so, do, 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 um, oh, Perfect. Okay. So today's meeting is for mainly all of you past participants, and we just want to share a lot of our recent findings from the studies that you guys participated in, and discuss them a lot more in depth. And really quickly, as more people are filing into the meeting room, we just want to say we're still recruiting and still looking for participants with SOS1 or other mutations. So if you guys know anyone who may be interested, we'll send the link in the chat right now, and you can distribute that link to anyone who may be interested. And there's a $10 referral bonus if anyone else signs up. So I'll leave that up for a second just for everyone to share it. And now we'll get started. So just to kind of run over the events, this meeting will be recorded, and we'll send out the recording after the event, along with some resources and some other details that we'll discuss in the meeting. And we'll address a lot of the questions you guys submitted in your form um, during the meeting, as well as we'll get to all the questions that you guys put in the chat at the very end of the meeting. But if you have questions during the meeting itself, please feel free to just drop them in the chat. And so just as an overview of today, we're going to go through two presentations quickly first from Dr. Tamar Green and Dr. Julia Plank. And then afterwards, we'll dive into the Q&A, which is the source questions that you guys submitted in the form. And afterwards, finally, we'll get to the questions that you guys are submitting live from the chat. So to introduce everyone, Dr. Tamar Green is our lovely PI here at Bridge. She's also a clinical psychiatrist at Stanford University. And then Dr. Julia Plank is one of our postdocs in the lab, and she's going to be discussing a lot of her work later today as well. And we wanted to also say thank you so much for participating with us in our studies and for the wonderful work and everything. Thank you. So.
Okay, so thank you, Aneta, and we're gonna get started. It's a really special webinar for us because it's the first webinar that we are kind of like sharing back the results from the studies that most of you here participated in. So we also kind of like want to hear back from you if that's helpful and what do you think about it. So welcoming everyone to kind of like drop in the chat or maybe write us afterwards. There is a QR code for our website, and at the website you can find all the webinars and okay, so I encourage you to kind of like use that. And I'm gonna do a quick plug-in again, is that our big study about Noonan syndrome is in like it's wrapping up. We kind of like anticipating to see participants in the next 6 months, but then close it. So we are in a search for participants with SOS1, Noonan syndrome with rare mutations other than PTPN11, but also PTPN11. We're looking for participants to do our online assessment. So encourage you to share with everyone that you can think of. That would be very, very helpful. The vision of the lab is to learn how altered genetics affect the brain and the behavior or the mental health manifestations. And in that way, we're hoping to learn a lot about the syndromes that we're studying, but also to contribute to the larger body of knowledge about genetics and child development, with the ultimate goal to improve the mental health and development of these children. Next slide. I just want to kind of like, again, introduce to our participants and whoever came to the lab, like a little bit more in depth what we do. So currently we have like 4 major studies. My guess is most of you participated in either the first one or the second one. And the first one is what Dr. Plank going to talk about today, is that we're looking into how can we learn more about the brain development that related to Noonan syndrome and NF1, and specifically we're going to focus on white matter. So this work comes from the first project here, analysis of neural correlates in rhizopathies. Also, the second one, we expanded in the last 4 years into studying also neurofibromatosis 1. So you'll hear results from that study as well, and other studies that are happening in the lab is looking at other genetics that can determine how children are functioning, meaning that we have the mutation either in the NF1 gene or in multiple genes in Noonan syndrome, but we also have other genetics that affects that. So we're interested in understanding that. This is number 3. And number 4, you actually participating in it now, in a way, is our efforts to extend our knowledge to the community and improving care through that. Next slide. Aneta, do you want the question?
Yeah. I was muted accidentally. But is there a reason that children with Noonan syndrome can have ASD but not be diagnosed at the typical young age due to atypical presentation?
Okay, so I really love this question, and I have to say that most of the questions you guys asked are actually a research question, so it's quite amazing how that aligns. And I'm going to answer this question using our published data from the recent studies. This study included 45 children with Noonan syndrome, and we compare that to controls, children without Noonan syndrome. And the way I'm going to answer the question is related to the fact that you see here that the children with Noonan syndrome, 35% of them that you can see here, presented with this sub-threshold symptoms of autism, but not with what we call MET criteria. And that explain the question and the feeling of many parents that the child is struggling with social communication and presenting with behaviors, but many times it's either not diagnosed or diagnosed later in life. Next slide. And this kind of like busy slide come to kind of explain that a little bit. So the definition of autism spectrum disorder is the core symptoms going to be kind of like divided into 2 groups: to social communication deficits, difficulties in understanding social interactions, understanding sarcasm, or understand kind of like interpret things really concretely and not like abstractly. So making gestures that helps with the communication. So that would be one group of symptoms. And the second group of symptoms is going to be a really restricted interest. For example, a child that likes to play only with airplanes and would not agree to play with anything else, or be very insistent on sameness. For example, doing the same routines again and again in the exact same way, like taking a shower in an exact same way, exact same order. So what we find is that children with Noonan syndrome and probably NF1 are presenting much more with this collection of symptoms and less with this domain, and that creates they do present with some of them, but not all of them, and that creates difficulties in the diagnosis. So many times children will present with some symptoms, but not the full blown, and that will create difficulties in diagnosing and even access to services. So that would be one of the reasons. I just want to kind of touch base that, as you can see, autism spectrum disorder, or let's say autism spectrum symptoms, can also come in association with irritability that we find really high irritability symptoms in NF1 and in Noonan syndrome. We find that many of the children present with the ADHD, anxiety, depression, less so for now because I think we're assessing very young kids, but autism do come with all this like spectrum of other behaviors. I'm going to stop here. Not yet, just a second. Netta, can you go back? I just want to kind of emphasize that in this study that I think Nomi just shared the link to, we did find several things that I want to point the listener to, is that we did find that children with Noonan syndrome present with this behavioral rigidity, difficulties in transitioning from one task to another task, and with difficulties in maintaining social relationships. So most of the children did suffer from some level of social isolation or difficulties to create this like strong social relationship. And also, as I mentioned, children with Noonan syndrome, in this case, in this study, presented with this elevated levels of irritability and emotional dysregulation, that both of them are considered domains that contribute to a diagnosis of autism. Next slide.
The next question was kind of a twofold question. It was, can these findings help with IEP processes, and how do I best help the education system accommodate a Noonan's child in school?
Okay, so I'm going to say it's a Noonan child and a child with NF1. So one of the recommendation that we can give is that we have this paper now available, open to the public. You can just print it out and give it to the school psychologist, because a school psychologist definitely should be able to fully understand the findings here. So that's our contribution to our scientific contribution to that it's proven, and Noonan is associated with those with ADHD, behavioral issues, and autism symptoms. Also at the bottom, this journal is kind of like producing a 5 minutes video. So if anyone prefers to listen to the results in a video, you can send them that, or even listen yourself. Next slide. And just to show a little bit of the results that how can we communicate and educate the teachers or the case managers, is in this study we evaluated executive function and attention, which are the cognitive abilities that contribute to diagnosis of ADHD. We also evaluated social skills that are related to autism diagnosis, and we evaluated aggression. And next slide. So here only if we focus, because of the question of autism, I focused only on social skills that we evaluated. And if you can, a way to read the results is that the red are children with Noonan syndrome, the blue are children who are typically developing. And you can see here quite consistently that children with Noonan syndrome will present with significantly elevated, in some places, even not almost non-overlapping, which means that the difference is really substantial, and show these elevated difficulties with, for example, communication, motivation for social interaction, and awareness of social difficulties, that in all these cases, the children with Noonan syndrome will present with more difficulties compared to controls. And I want to add here that we in a statistical manner, we controlled for the fact that some of the kids with Noonan syndrome also have some effect on their IQ and their cognitive functioning. And we were able to show that this is not related to their cognitive functioning. It's actually what we believe a core deficit that's related to Noonan. Next slide.
Mhm.
And just to wrap up with that, about the question about how we can help with IEP processes, first of all, is understanding them better and learning how to communicate with school. And I want to kind of direct you to another resource that we have, is this webinar that is utilized over 30 years of experience from educational advocator at Lucille Packard Children's Hospital. And I think Tahar did like a great job explaining these processes. I am a child psychiatrist, learned a ton from her. So I recommend to watch that. And it's really kind of like has this step-by-step how to communicate about an IEP.
And then finally, the next question was, how do brains of people with Noonan syndrome function differently or similarly to typical peers? And we're going to transition quickly here to Dr. Julia Plank, who's going to dive more into her work and also answer the question throughout. So Julia, if you want to also screen sharing, if you want to take over.
Cool. Let me do that now. Cool. So I have to say I'm a little bit nervous presenting because the questions that everyone sent in were so fantastic. I feel like I'm speaking to a room of experts, but I'll do my best here. So I'm going to present here specifically on MRI findings. So I know for your children who participated in the MRI scans, they're very noisy. They can take a very long time. They're boring. They can be claustrophobic and, of course, have to remove metal. They're a bit of a hassle. So I know they're not the most pleasant experience, although I personally have a love for MRI and analysis. So I wanted to explain why we use MRI and what we can find, some of the findings from MRI as well. So MRI is basically a large magnet with additional smaller magnets that applies magnetic fields. And from there, we can take really cool images of the brain and of the body. We're specifically focused on the brain because of the link between brain and behavior. And MRI has many benefits. It's not invasive. It doesn't use X-ray like a CT scan. It doesn't use radiation like a PET scan. So it's an extremely safe form of imaging, and it gives you really beautiful images. So when we were asking your children to stay really still, it was so we could get beautiful images like these, and we can see really nice gray and white matter tissue contrast. So this is an image like a bird's eye view of the brain. If you chop off the top of the skull and you look directly in, we can see the ventricles here. So we get these really nice images. And from there, we can find out all sorts of things about brain volume, the thickness of the cortex or the outer layer of the brain, the orientation of different fibers in the brain, the connections between brain regions, and many more different things as well. So what I'm specifically talking about and what I'm interested in is brain microstructure. So this is the microscopic structure of the brain. So rather than looking overall at the brain as a large volume, I'm looking at the cells in the brain. So diving in really deep to look at the presence of different cells, their densities, how many there are, and the alignment, their orientation. And so this is an example of just some of the cells that are present in the brain. So we have the neuron, which is sort of the main brain cell or the nerve cell. But there are many other different cells as well, which I'm not going to explain all of the different ones. But essentially, every cell in the brain has multiple roles to play. They have different functions. And so when there are more of certain cells present, or when the cells are structured in a certain way, we can find out about brain development. We can find out about whether there's an injury that's occurred, if there's inflammation in the brain. So understanding how the cells are structured in the brain and their densities can help us understand a lot about what's going on for your children and in Noonan syndrome and in neurofibromatosis type 1. But MRI is limited in that it can't actually visualize individual brain cells. So these brain cells are only about 0.1 of a millimeter is their average size. So they're really tiny. There are 86 billion of them in the brain, neurons alone, excluding the other cells. So we have to use a proxy measure, some other way of understanding how the cells make up the brain. And what we use to do that is water. So about 80% of the brain is water. So there's water throughout the brain everywhere. And water works so well, not only because it's so abundant, but also because of the makeup of water. H2O has these 2 hydrogen atoms, which actually have inherent magnetic properties, which means we can image them really easily and really well with MRI. And we can use water to understand cellular structure and brain microstructure, because we know that water moves differently depending on different brain tissue types and different brain anatomy and different cells that are existing in the brain. So, for example, in the ventricles of the brain, which are here in the center, they're filled with cerebrospinal fluid, or CSF. And we know in here that these water molecules can move really freely. For example, it's as if they're like in a bucket of water or in a pool of water. You can imagine the water molecules moving really freely in different directions. However, for example, in a neuron, water is more like in a narrow pipe. So maybe it can only move in one direction or in 2 directions. It's much more restricted, and we can actually image this movement of the water using MRI. And so based on these principles, we can get a really deep understanding of the presence of different cells in the brain and the structure of them. Whoops. So now that we've sort of got that background out of the way, what I specifically have been looking at recently is the brain tracts. So these are like the highways that connect different brain regions. And within each of these tracts, there are different cells, different microstructures. So I'm looking at the microstructure of each of these brain tracts. And thanks to all of your children who participated, so we had 31 children with Noonan syndrome come through the MRI, 25 with neurofibromatosis type 1, and then 32 typical developing children as well. So we were able to compare the microstructure in these brain tracts between these 3 groups. So this is a lot, so I'm just going to go through it slowly as much as possible. So here we have the 39 different tracts that we looked at in the brain in each of these groups of children. So in blue, we have typical developing, in orange, Noonan syndrome, and in pink, neurofibromatosis type 1. And down the bottom here, this mean represents essentially the density of cells in each tract. And so what we can see is a really clear pattern where the cellular densities in each tract seem to be consistently higher in typical developing children compared to children with Noonan syndrome and NF1, the children with Noonan syndrome and neurofibromatosis type 1 have generally very similar cellular densities, except in this tract here, the middle cerebellar peduncle. So this is the only tract where we could see a really clear, significant difference between Noonan syndrome and neurofibromatosis type 1 in terms of their cellular density. So this tract, the middle cerebellar peduncle, you might not have heard of it before. It's a tract that is at the back of the brain. It runs through the cerebellum, which is a brain region involved in motor coordination and balance. It's involved in a lot of different functions. It's a really important brain region. We know that particularly in neurofibromatosis type 1, the cerebellum is prone to hyperintensities. But this tract, the middle cerebellar peduncle, has not really been highlighted as a tract of interest in the research before. So we're very interested to come up with this finding. So why are these results important? What do they mean? So in this work, we were able to identify really clear microstructural differences between the typical developing group and between Noonan syndrome, neurofibromatosis type 1. And the pattern that you saw is really quite remarkable. They're very significant findings. And of course, as I noted, we did see similarities between almost all of the tracts in terms of the cellular densities in each tract. So similarities between Noonan syndrome and NF1, which is really somewhat unsurprising, I suppose, given that they are both rhizopathies, so they both have the same genetic pathway that's been implicated and some overlapping clinical characteristics. But we also know that there are many differences between Noonan syndrome and neurofibromatosis type 1, which is why we were really interested to see that one tract, the middle cerebellar peduncle, showed really clear differences between Noonan syndrome and neurofibromatosis type 1, and that's a tract that we'll be looking at in further investigations. So as I mentioned, we'll want to, in future work, look further at this particular tract and try to understand what it means that it's comes up as showing significant differences between Noonan and neurofibromatosis type 1. And we'll also want to look at if there are any associations with clinical characteristics as well. So as Dr. Tamar Green said earlier, what we're really interested in is the link between what's happening in the brain and the link to behavior. So this is really just the first step that I've presented today, but we want to see if there are associations with clinical characteristics. And then down the line, we're hoping that perhaps we can identify something in the brain and the underlying biology that might act as a potential target for treatment. So most of the treatments at the moment, as you would know as parents, are focused on the symptoms and they might not be so effective. And so we're hoping by understanding more about the biology that we can eventually find more effective treatments that target the underlying causes in the brain. And finally, I just really wanted to say a big thank you to all of you and to all the children who participated, particularly in the MRI section, which I'm involved in because I know it's often probably the children's least favorite part, but I really appreciate them coming in and persisting through it. So thank you so much.
Thank you so much. We're going to jump into the Q&A from the source questions from earlier. And then if you guys have any current questions, you can put them in the chat and we'll get to them right after the Q&A questions. So to start, how can you tell a behavioral connection like Noonan is more easily irritable when each child is born with a certain degree or lack of irritability tolerance? How do you know it's a Noonan syndrome connection versus a natural variance in a child?
Okay, so I'm gonna start. As I said, like these are research questions for no doubt. So first of all, thank you for the question about irritability because this is something we learned during our I learned in clinic, seeing a lot of children with Noonan syndrome and maybe rhizopathies in general, is this reports about irritability. And the question asked, you can ask this question about any behavior, right? Like when do you know it's related to Noonan, and when do you know it's kind of, this is, something that is maybe within the norm or outside of the norm, but not related to Noonan. So for the question whether it's related to Noonan or not, the way we test that is by comparing our groups, as you heard from me and you heard from Julia, that we compare the groups to typically developing controls. And this is our scientific approach to show that at the group level, as a group, children with Noonan present with more irritability. And just kind of like to inform the audience that this is one of the studies that Dr. Yaffa, that I see here on the call here, also working on. So the way to know if it's related to Noonan or not is comparing that to controls, but eventually we deal with the individual child, right? And we are managing the irritability in the individual person. And there, I would say from a clinical perspective, currently, I would say we need to treat the irritability as it doesn't matter, right? It doesn't matter if it's related to Noonan or it's a natural variance in the child. When it's gonna matter to us is at a point that we'll have specific interventions that, let's say, target the MPK pathway or target specific alteration in Noonan syndrome. And then we want to know whether a behavior is related to Noonan or not. I think at the individual level, it's not an easy question. And as a physician, knowing a child has Noonan syndrome and presenting with irritability, my first approach would be to assume that it's related to the Noonan syndrome. Finally, and I hope I'm not complicating things too much, is that, as I mentioned at the beginning, that we have this project that also looks on the other genetics. So today in genetics, we do have a way to measure the risk for irritability, for example, or the risk for ADHD symptoms, attention symptoms. So ideally, in the future, we can imagine a time that we kind of have this knowledge about the Noonan syndrome, but we can also measure the other genetics and have a more complete understanding of the specific child and their risk to present with irritability behavior or other behavior.
Absolutely. Yeah. And moving on to the next one, someone asked, could you elaborate on differences in social cognition and social interaction in children with Noonan syndrome?
So I'm trying to find the best kind of like way to think about it. I will almost say that social cognition, in a way, it's not a perfect example, but social cognition would be the theory that if you learn how to drive, social cognition will be the theory of how well you can understand. If I present to you with two children and I ask, like, one of them seems like angry and one of them seems like defensive, you're able to describe to me what is the social interaction, right? The same thing like when I test you in theory before in the DMV before you actually learn how to drive, whether what does its stop sign means. So that kind of like more into the social cognition, while social interaction is more as the driving, right? Like what happens in a real world situation with other real children or with other real individuals. And of course, if the social cognition is affected, we can expect that the social interaction might have also going to be affected, but not always, right? Like sometimes sometimes it's possible the social cognition is intact, but when we actually the child is actually in the world, we see that there's a lot of issues, social interactions. But then we sometimes think whether other causes kind of like affect that. For example, if the child is extremely inattentive and extremely hyperactive, so there's going to be an issue with social interactions, but not necessarily it's going to be related specifically to the social cognition, if it makes sense.
Yeah.
It's kind of like then, like if someone cannot drive, it's maybe because he doesn't have like good glasses, right? Like maybe it's another reason.
Fair. Yeah, all about the tools. And next up, someone asked, are there therapies that target executive function delays in particular?
Yeah, so I would say yes, there are. There's several, and I can like do some to drop some resources, but there are interventions that help the child mostly from the aspect of organization. Organization is also considered, executive function is a big umbrella. So there's a lot of guidance, like books that guide how to have those interventions. There are also a lot of executive psychologists or experts that work with a child on these aspects of executive function in terms of like time management, planning, assessing whether like stopping for a moment and assessing your work. So it's mostly behavioral training interventions that improve this functioning.
Yeah, absolutely. And does the amount of white matter or lesions found on a brain MRI with someone with NF1 correlate with the severity of learning or behavior issues? And I guess this one goes more to Julia.
So there is some evidence suggesting that this might be the case, but the research at the moment is kind of currently unclear, and so we don't know for sure, and we really need some longitudinal studies to find out if white matter, the amount of white matter or lesions on white matter might correlate with learning or behavior challenges. So we don't know at the moment. Possibly.
Yeah. And the next question, were there any IQ correlations between the parents and the participants? And following that, how about the psychosocial questions? Were they similar between the parents and the children?
Yeah, so I actually have like almost ready answer for that because this is a paper that is already ready and got evaluated, and we just like editing it. So I think our next webinar we can expand on that, but just to give a yes, we do find a correlation between the IQ of the parents and the participants in a way that we what we think that happens is that I think about it like height. So let's say that we have two parents, one of them is very tall and one of them is shorter, and there's a way to kind of calculate the child expected height. So it's not exactly the same, but there is some evidence that shows that naturally there is a correlation between the parent's IQ and the child. And it's the same thing for children with Noonan syndrome. I can speak less to NF1 because we didn't do the study there, but Noonan syndrome has this like effect on IQ that on average there's a loss of 10 points of IQ. With that being said, I just want to remind everyone that IQ is a very specific measurement of a very specific abilities. And in a child, there's so much more to that can affect them being able to thrive. So I just kind of encourage everyone not to get stuck on that. And about the other part of the question about the psychosocial, so we see the similar kind of like trends, that there is a correlation. It's not a perfect correlation, but there is a correlation. Part of it might be because of genetics. Part of it might be also because the environment, because if a child grows up in a I will take anxiety, for example. If I'm a mom who has anxiety like tendencies, my child is growing up in an environment is more anxious and has more chances to model these behaviors and be anxious themselves. So
Yeah,
yeah, hope it makes sense.
that's fair. And also, how will your findings drive further research? And this also kind of ties back to Julia was saying in variant of representation.
Yeah, I will let Julia answer that.
Yeah, so I guess sort of to reiterate, well, yeah, I think in several ways. I mean, I can mainly speak to the findings in the brain. So we're trying to find differences in the brain that exist between typical developing children and then children with Noonan syndrome and children with neurofibromatosis type 1. And then we're trying to see if these changes in the brain are associated with patterns of cognition or thinking or social skills, and what's the relationship between the findings in the brain and these different clinical characteristics or behavioral characteristics. And then following that, we hope that eventually that will lead to development of more targeted treatments that can target these brain regions or differences that we find in the brain. And so they're more precise treatments with more precision, which means that hopefully they'll be more effective for children and alleviating their behavioral symptoms as well. I don't know if you wanted to elaborate any further on that, Tamar.
No, I think you did a great job. I do want to say one thing, but I'll see the next question to kind of like see.
Fair enough. But yeah, absolutely, and especially with the lab and our vision, kind of just looking at the cause rather than the effect a lot of the time. Actually, that was the last question
Yeah,
before we
so
jump to the open floor.
I'll just add to that.
Yeah.
Just hearing kind of like Julia and seeing other lab members is just that I kind of like also want to share and say thank you to our lab members, because I'm not sure how much people are aware that working in academic medicine is not the most financially rewarding, and it's a very long career and very it's pretty hard work. So I also want to kind of acknowledge the efforts that our team is doing. In addition to huge thank you for everyone who participate in our studies. It definitely help us to go back to funders and say that we working with community that is very kind of like engaged. So yeah, so just be thankful for that.
Yeah. We only have a couple questions and we only have a couple minutes, so we're gonna go through these kind of quickly. One of the questions was, are there any known treatments or medications that seem to decrease volatile behaviors or irritability in kids with NF1 or Noonan syndrome?
Decrease, I didn't hear, I heard the irritability, I didn't hear the other one.
Volatile behaviors.
Volatile. So usually when you assess a child, we try to get to some what we call a DSM diagnosis. So and kind of like find the kind of maybe the kind of like exact square that we can think about these symptoms. So irritability and volatile behavior can, what I'm trying to say is there can be many reasons for that. So I would say that if I think that the core symptoms are inattention and hyperactivity, so there's medications for that, that will be from the group of stimulants. But if the child is very anxious and irritable, so I would think about medications like SSRIs. And I jumped immediately to medications because I'm a child psychiatrist, but I just want to emphasize as well that usually first line going to be behavioral intervention and understanding what's happening in the family. So not all the solutions in the treatment are medications. There are also many other options.
Yeah. Our next question is, someone asked, is there a correlation between the extremity of a child's physical symptoms and the extremity of their behavioral symptoms?
Oh, that's a great question. From my knowledge, I am not aware of these correlations in rhizopathies, meaning like Noonan syndrome and NF1. There's some work in other syndromes that show some correlation between cardiac effect of the syndrome and some manifestations, but I would say that typically it's not as you would expect. It's not like very clear correlation. So sometimes we can see kids who are like, let's say the penetration for the heart is like really low, but the behavioral manifestations are much more severe. So I'm saying that with like, I did not review the literature really deeply, but that's kind of from the general knowledge.
Yeah.
Tamar,
Yes.
I just read an article like 2 days ago, but again, it was like
please.
that they found no correlation with heart problems in behaviorals. But again, maybe it was like
in which syndrome?
I need to remember well. I think it was rhizopathy, but now you put me on
okay.
But yeah, I do remember that, and I wanted to. But
the finding that I was referring to
22Q.
22Q.
Yes, maybe it was 22Q. You're right. Sorry.
Yeah. We're running overtime, so we'll do one last question before we kind of wrap up. But someone asked, do the differences in brain densities between kids who have NF1 or Noonan syndrome and typical kids correlate to differences in processing and behaviors?
So my understanding is do the differences that we found in cellular densities in the brain tracts correlate with behavioral findings? So at this point, I would say not convincingly. I think correlations are a tricky subject. Statistically, they're sometimes not the best way of understanding links between brain and behavior. So actually at the moment in the lab, we're working on another method of trying to predict behavior using a more rigorous and robust statistical method based on those brain imaging findings. So using those findings in the brain to predict behavior in a more robust way. So the correlations we found were not significant, but I would say there's more to come on that. So maybe stay tuned.
Okay, interesting. And on that note, we're going to wrap up. We wanted to really quickly provide more resources. A couple of these organizations provide really great resources for connecting parents and helping parents in various ways. And we also, there's a couple more here. A lot of these address different aspects and different ways of helping. And we wanted to thank a lot of these foundations and networks. Some representatives are present today, just for distributing our studies and all the work that they did. We have, once again, if you guys know anyone who would be potentially interested in participating in a study with us, especially if they have an SOS1 or another mutation, please feel free to refer them. There is a $10 referral bonus. And we are happy to provide more information if they reach out. And in general, thank you so much for participating in the study and for joining us today. We'll send a recording after all of this, along with all of the information, all the resources that we mentioned today. So thank you.